I'm absolutely thrilled to be here and to see you all gathered together for an evening of, I hope, fun and a little bit of science. I'll keep it at a low level. But I also should say that I'm thrilled to be here at the Rockefeller. I've been here for 27 years. And even though I love telomeres more, I love the Rockefeller a whole lot. And it wasn't a crush. It was sort of a forced marriage. But, you know, they can work. And I really love this institution. Where else do you see your friends do great science, and then eventually you wake up on a Monday morning and they've won the Nobel Prize? Only at the Rockefeller. I have 5 Nobel laureates in my building. So let me tell you a little bit about the work that's going on in my lab. What I'm going to talk about is the cancer-relevant aspect of our work. I will also tell you a little bit about telomere biology as it comes into this story. I'm basically going to tell you what happens to the DNA in our cells when cancer develops. And it's something that has puzzled me for a long time. And it's a scary subject, of course. We all hate cancer, nobody more than me. But it's quite interesting what we've learned, and there's a lot of good news in this story. So we're going to talk about the genome, which you know is a set of these boring 4 letters, A, T, G, C, that together make up the genetic code that determines how our cells behave. So the long DNA in our chromosomes, it's very, very long, contains an endless numbers of A's and C's and T's and G's that make up the genes that encode the different proteins that your cell needs to build itself, to build all kinds of things, to make small molecules. Some proteins come together and form building blocks themselves. Some proteins degrade other proteins, and so on and so forth. So these genes are, of course, differentially expressed. There are different genes that are on in your brain cells, whereas in your liver cells. There are genes that are on, I hope, right now, trying to take care of the alcohol you just consumed. And the brain cells just feel the alcohol but can't remove it. So all this DNA is incorporated in an organelle that's called the nucleus that is stained blue here. And each of our 10 to the 13 cells, I don't even know how much that is. Probably some business people here are better at those numbers. To me, that's just a gazillion. We have a gazillion cells in our body, and each cell contains in it the 46 chromosomes. You know from 23andMe, get 23 from your father and 23 from your mother, together making up the 46 chromosomes in our genome, and here you see them. And they've just been duplicated. The DNA has been copied. So each chromosome is present in 2 copies that are still held together, because this is right before the cell divides. So here's a blow-up of a chromosome. You see it's held together there, and when a cell divides, it will bring one half to the daughter and the other half to the other daughter, and so it segregates the genetic information with high fidelity. This is a large chromosome, and that's a tiny one. That's chromosome 21. So I have been studying chromosomes for the greater part of my life. I first saw a picture of chromosomes when I was in high school in Holland, in Rotterdam. And I saw this picture. It was not this picture, but it was pretty similar with this chromosome, and you can see how much DNA is in there. And it's just mind-boggling that it's all organized and wrapped up. And I really thought it was very interesting how the cell manages to do this. So I have worked on chromosomes for a very long time. And as this happens to scientists, and this may sound odd, but if you work on something for a very long time, you start thinking it's beautiful. It's sort of like every parent thinks their child is unbelievably beautiful. Most scientists believe that their subject is just beautiful. And so when I think about chromosomes, I see something like this. Beautiful. Here, Lange has painted this young woman, hopefully not as young as, I won't mention Roy Moore, shall I not? He has painted this woman. He's done a great job, as he always does. You know, the part of the painting is out of focus, and then you're drawn into the eyes that are in focus. It's just beautiful. It is balanced. It is functional. You know exactly what it is. And in contrast, when I think about cancer chromosomes, which are changed during the process of cancer formation, I think of something like this. This is a painting of a woman by Willem de Koning, who was also born in Rotterdam, just like me, worked here in the US. And it clearly still is a woman. You can see that he is painting a woman. It's an impressive work. All the bits that sort of define women are still there. But it's not quite normal anymore. Things have been rearranged. And you wouldn't think of it as beautiful. It's impressive, but not beautiful. So that's how I view these two genomes, the normal genome with a lovely set of chromosomes all paired up, all identical, and the cancer genome, which is really distorted and altered. So if we zoom in on the cancer genome, this is just an example of one particular cancer cell line that's been analyzed with a technique whereby different chromosomes are colored with different colors. And so you can see that the yellow chromosome 1 is there in 2 copies in a normal cell. But in this cancer cell, one of the 2 copies is just a very large piece of another chromosome on it. And this is called a translocation. And then if you look at chromosome 4, both copies have a translocation. They're both altered. And for instance, if you look at chromosome 19, the chromosomes are numbered according to their size. So you get smaller as the numbers get bigger. If you look at 19, something very odd is happening, because instead of 2 copies of this chromosome, there are now, 6? And many of them, again, have unusual changes in the chromosome. So this was known when I started thinking about this problem. How does this change occur in the cancer genome? And it's seen in many cancers, not in all, but in many cancers you see these kinds of changes. Changes in the number of chromosomes. Often the number is no longer 46, but closer to 90. And changes in the structure of the chromosomes. When I started thinking about this process in the early 90s, there was a general model in the field of how this might happen. And the model was simple. It proposed that there was a gradual accumulation of chromosome changes. So you start out with a Lange, cells divide, one cell becomes somewhat tumorigenic, but isn't there yet, and undergoes one change in chromosome 1, for instance. And that cell then proliferates and makes a little nodule, and one of those cells undergoes a second change, chromosome 4, and a third change in chromosome 19, and so on and so forth, until you end up with this completely rearranged genome of the de Koning. Now, this was a good model, and it may well hold for many situations, but the problem is that in order for this model to work, there has to be a certain frequency by which these changes occur. And if you calculate it out, you don't quite get there. Of course, cancer takes a very long time to develop, 10 years, 20 years, and involves many cell divisions. But even if you take all that into account, it's hard to get at a completely rearranged genome with these individual small steps. So I started considering that something else might be going on. And the idea was that perhaps for the longest time, nothing happens in cancer to the genome. We stay with the Renoir picture, and then something dramatic happens, an explosion in the genome that leads to all these changes. And the reason why I decided that this may be what is going on is because we were working on telomeres, and we knew that the telomeres were disappearing. And that disappearance of the telomeres could, in my mind, be held responsible for creating this change in the genome. Now, I'm going to introduce telomeres to you, which are the love of my life. Here they are. Each chromosome, each DNA strand has at each end a telomere. You can see them here, highlighted with a FISH probe, a hybridization probe, that detects the telomeric DNA. You can see in this human chromosome spread that every single end has a telomere, and it better have a telomere. So what are these things? Let's zoom in this chromosome that we'll pull out and look at it in detail. And if you look in detail what's going on at the end of this chromosome, you will find that instead of having a mixture of A's and T's and C's and G's, you'll find that this DNA at every chromosome end is composed of a repeat sequence. It goes TTA, GG, TTA, GG, TTA, et cetera, et cetera. Some people think this is named after my name, or vice versa. Coincidence? TTA, GG. But it's a pretty boring sequence, and there's a lot of it. We are born with thousands of copies of this sequence at our chromosome end, enough to get us through a lifetime. So this DNA is at every chromosome end exactly the same, and it's in every cell, and in fact, every vertebrate, every reptile you would look at has exactly the same sequence at their chromosome ends. And that is because the function of this DNA is critical to the viability of cells and organisms and species. So what does this DNA do? Well, it helps out with an intrinsic problem that our chromosomes have and our cells have. Our cells have a very good way of looking and monitoring their DNA for damage. We like to keep our genome intact, especially in cells that otherwise could give rise to cancer. Our cells invest a lot of energy in keeping the DNA intact and in the germline so that our children get perfect DNA. And what happens when there is a break in the DNA or some other lesion is that cells take note. They can see the DNA break and activate an alarm system. So there are proteins in the cell that can go and find these broken ends, and then they activate what is called the DNA damage response, which does two important things. It, on the one hand, prevents cells from dividing. So the alarm goes off and it says, stop construction, because there's something really wrong here, and we need to first figure out what is wrong. And at the same time, cells endeavor to repair the break. So once then the break is repaired, the alarm system withers away, it's switched off, and now cells can go into cell divisions again. And this happens every day in all your cells many times. There's a break, it's detected, cells go, ooh, let's stop for a bit, it's repaired, and then the cell goes on and can divide again. Now, since this alarm system can find one DNA break in the DNA of 46 chromosomes, it needs to have some way to know that this is not something that one should be concerned about, because the telomere, the end of the chromosome, is just a DNA end. At the molecular level, it just looks exactly like a DNA break. So cells with linear chromosomes had to evolve a way to allow this alarm system to make the distinction between a DNA break somewhere in a chromosome and the natural end of the telomeric DNA. And that's of the chromosome. And that's what the telomeric DNA does. It allows cells to make that distinction so that the alarm system doesn't go off at the ends of our chromosomes. So how does it do that? That was the puzzle that I started working on 27 years ago, and I still haven't solved the puzzle. It's embarrassing, but we've learned a lot on the way. And the major trick we found by which cells learn to ignore this type of DNA end is by actually hiding the DNA end. So the telomere is actually not a linear thing with an end somewhere, but it's a loop. We found this by looking through the electron microscope at human telomeres that we had purified in collaboration with Jack Griffith at North Carolina, University of North Carolina. And what we found is that the telomere, so this is the end that goes to the chromosome, it's been cut off there, that the telomere does not have an end. The end is tucked in. And so this is why the cells know that that's a telomere, because they just can't find the end. There's nothing to alarm the cells. There's nothing that the DNA damage response can find there at the end of the chromosome. So then the question is, how does that happen? How do you get this T-loop, this looped conformation at telomeres? And we found that that's the job of a protein complex that we called shelterin, because it shelters the chromosome end. And it's shelterin that binds all along the telomeric DNA and tucks in this end of the chromosome right there and hides the chromosome end from the DNA damage response. So this is how it works. You have telomeric DNA, a loop back, you hide the end, everything is fine. And it's because of this that some people are very concerned about losing their telomeric DNA. So as you may know, the telomeric DNA in our cells slowly withers away. Every time one of your cells divides, there's nothing you can do about it, the telomeric DNA gets a little shorter. And the basic problem why this shortening occurs is the problem of DNA replication, which just like when you paint a floor, you can't paint under your feet. DNA replication cannot finish a job at the end of a linear piece of DNA because it can't replicate the DNA under itself. So as a consequence, our telomeres shorten. So this is a graph. It's not my work. It's from people at NIH. It's a graph of the length of that telomeric DNA. You start out with a lot at birth. It's thousands of copies of that repeat. And then over the aging process, it goes down. And it's true, although there's this enormous cloud of dots here, overall you can see this trend of it going down. Now, I'm sure there are some people, not me, who, you know, in this audience who have very long telomeres for their age, and some people who are young and they have shorter telomeres. I think I'm basically out here somewhere. So telomeres shorten, and a lot has been made of this. And there is a view that this is a way to predict your biological age, and there's even a company where you can send your DNA, and they'll tell you how old you are according to this graph. Now, I'm not promoting this company. I don't have any financial involvement in this company, and I'm not sure you could actually predict much from this cloud of dots. And so if you want to know your biological age, my advice would be just look in the mirror. Works for me. It's free. So the loss of telomeric DNA is just something that happens like our hair goes gray. But why does it happen? Why did biology set it up this way? Why don't we have a system to maintain the DNA? Well, that's because this loss of the telomeric DNA with cell divisions is actually really useful in our species because it blocks tumor formation. So every time a cell divides, some of the telomeric DNA is lost because of this problem. And this happens in our normal cells, but it also happens in cells that are on their way to forming a cancer. And in those cells, it's a way to keep track of the number of cell divisions the cell has been going through. The system is set up such that if the cells have divided a certain number of times and their telomeres have shortened to a specific setting, which is the alarm setting, an alarm goes off, and the alarm forces the cells to die. And it's a great system. In our species, we need very strong tumor suppressor systems like these in order to be as long-lived as we are, but especially in order to breed at a very late age, 12 or 13, the official breeding age when our species becomes fertile. That's an unusually late age in any animal. If you ask a mouse, right, they become fertile in a few weeks after birth. So 12 years seems short to you, but it seems very long for a mouse. So we need extra tumor suppressor pathways to keep cancer out of our species for as long as we need to breed. And mice, for instance, don't use this system. They get cancer very frequently at the age of 1 or 2. So imagine if we didn't have this, what would happen in our system? Now, like any tumor suppressor pathway, occasionally it fails, and there is a cell that doesn't pay any attention to what's happening, and it continues to divide, and the telomeres get shorter and shorter, and then there is an entry in a stage that we refer to as telomere crisis. The telomeres are now so short that all hell is breaking loose in the genome. But again, this is not bad, because it's another way to stop the now further progressed early stage of cancer, for instance, DCIS, the early stage of breast cancer, to stop dividing further. Because when telomere crisis occurs, the whole genome is so unstable without this telomere protection that, again, the cells die. Now, finally, very rarely, but unfortunate for those of us who have cancer, it does happen, a cell even breaks through that second block, and that is through the activation of an enzyme that can add back the telomeres. This enzyme is called telomerase. It was given the Nobel Prize 10 years ago, and this enzyme is normally only active in our germline and stem cells, where we want to maintain the telomeres for our progeny. But cancer cells have found a way to get around the problem by activating this enzyme in very sneaky ways. The enzyme then elongates the telomeres, and now the cells can grow again. They're out of the crisis. They have escaped. But cells that have escaped crisis have genomes like this. And we know this from work that we have done in my lab to model the episode of massive genome instability that occurs during telomere crisis. So what we've done is taken cells and we let them grow under certain conditions into this stage where things go terribly wrong, and then we can directly observe how you go from a Renoir to a deconing. So one thing that we found is that when cells are in telomere crisis, the chromosomes are no longer separate. They start fusing together. And the cells then try to divide and pull these two chromosomes apart. And what happens is that a break occurs, and that then gets fixed, and you get these translocations. Another thing we found, a graduate student in my lab found that when cells are in telomere crisis, they make terrible mistakes in their cell division cycle. Rather than duplicating their DNA and then dividing, they duplicate their DNA twice. So you go from 46 chromosomes to 92. And that can explain why we see these high copy number of chromosomes in cancer. And then finally, in work that we published just 2 years ago, we found that during telomere crisis, some very exceptional events occur where chromosomes get shattered into pieces and put together in inappropriate ways. Okay, so what have we learned? We have learned that during tumorigenesis, especially of solid tumors, telomeres shorten. This is good because it blocks tumorigenesis. But when these blocks fail, it can create the coning-like genome. But even in that sort of disappointing aspect of the story, there's good news. Because we think that the presence of these alterations in cancer can have certain diagnostic potential. So we are currently looking for a signature in the cancer genomes that can tell us whether the cells have gone through telomere crisis. And once we have that signature, we have one now, we will go to early cancers and ask, has this cancer already gone through this terrible moment? And if not, perhaps it's more appropriate to monitor that particular tumor to see whether it will regress, as it should, in telomere crisis. Whereas if you find a tumor that has already gone through crisis, it's more likely to progress to a full-blown cancer. So in addition, we think there may be therapeutic implications because the coning-type genomes are more likely to have encode new proteins, and it's the new proteins of cancer cells that are recognized by the cancer immunotherapy that was recently developed for melanoma. So we're very keen to look into this. And then finally, we have the view that probably the cells in this stage that have gone through telomere crisis have something akin to cellular PTSD. They've had a crisis, they had a traumatic experience and probably have been altered, and perhaps therefore we can find drugs that will kill these cells but not normal cells in the body. So with those thoughts for the future, I will stop and say again how wonderful it is to do this kind of work at the Rockefeller. It is not only an oasis as depicted here in terms of landscaping, but it's an intellectual oasis where you can just be away from the rest of the world, and it's really wonderful. Also want to thank the funding agencies, but most importantly, I want to thank the people in my lab who are just terrific. And I only make their life harder. Thank you very much.
Thank you, Titia, for that spectacular talk. I'm sure there are questions from the audience, so if you have a question, raise your hand. We have roving microphones, and so a microphone will make its way to you. And please keep your hand up so they can find you. I'll start the questions and ask Titia, you mentioned that some of these chromosome rearrangements may create new genes that are contributing directly to oncogenesis. What is the current thinking about how these cancers develop in the setting of these massive rearrangements? Are there particular drivers that are made by the rearrangements, or is it just the abnormal number of chromosomes that is some way contributing to the development of cancer?
Well, the frequency of the changes that you see leads to a greater chance that one of the breakpoints where one chromosome is fused to another will contain an alteration in a gene that can help the cell grow or help the cell evade the immune system or do something else that will lead it to be more cancerous. So in many of the cases, the changes that are seen in a genome are not at all associated with some change in a gene. But there are very good examples where such changes, in fact, do drive cancer.
Yes, great presentation. So I'm wondering, what do you think is causing the, well, we know it's causing the telomeres to shorten because of aging, right? But what do you think is causing the chromosomes to shatter like that? Is it something biological inherent in the cells, or is it something environmental, man-made chemicals, and so on?
So this is a question from one of my colleagues. There you go. And so I will not answer the question completely. You can look up the cell paper. We have figured out the mechanism, But basically what happens is that when cells try to divide with these fused chromosomes, they start pulling and pulling at them, and then an enzyme gets confused on this pulled DNA and just cuts it into pieces. So it's not a normal phenomenon. It only happens in cells that are in crisis, but it drives the shattering of chromosomes.
Yes, that was a fascinating presentation, but I'm just curious, what is the takeaway as to what we can do to prevent that from happening?
Prevent what? The telomeres from shortening? Yes. Well, my argument would be that you really don't want to prevent it, because the shortening of your telomeres is one of the best things you have. So embrace it. If you wanted to have longer telomeres, think about it. This system blocks tumorigenesis, the shortening. So if you start elongating your telomeres, what are you doing? You're giving cancer cells a better chance to grow out. And, you know, none of us think, you know, maybe a wrinkle less is worth the risk of promoting cancer. I also should say that the evidence that telomere shortening actually has physiologically important effects in the human system is minimal. There's some diseases, childhood diseases, that are due to short telomeres. It's very sad. These are children that are born with telomeres that are much too short, and they have too little of the telomerase to heal them. These children suffer from progressive bone marrow failure. That's their main symptom. And they get transplanted, and then they do fine, and then they get lung fibrosis in some cases and liver cirrhosis in some cases. Now, if we think about human aging, what diseases come to mind? Cardiovascular disease, Alzheimer's, Parkinson's, etc. None of these phenotypes are seen in these children with very short telomeres. So that is a genetic argument to say that it's not that likely that we have Alzheimer's because our telomeres shorten. I wish it was not true because I could get Alzheimer's funding money. Bill Gates just gave another $100 million, but there it is. It's very unlikely that telomere shortening has an impact in the cardiovascular system, in the central nervous system, and so on.
You mentioned funding. Is it harder to get funding for the kind of science you do than it is to get funding for Alzheimer's? I mean, I hear Alzheimer's scientists sometimes complaining they can't get funding either, but just curious.
Well, I don't want to spoil things for my esteemed colleagues in the neuro arena. It's hard for everybody, but you know, we basic cell biologists often joke, you know, if my process was only relevant to a neurodegenerative disease, I could get that money. But then the neuroscientists joke, if my process was only relevant to cancer, I could get that money. So it's not easy these days to get funded. It wasn't easy when I started in 1990. It was very hard. There was a period after Harold Varmus had increased the NIH budget when it was a little easier. Now it's very hard again. It's just very hard.
Thank you, Titia. I was just wondering, are there any particular variables in the environment that particularly affect the health of telomeres?
Yeah, there is one major problem in the environment that if you get too much of it, your telomeres will bite the dust, and it's oxygen. So don't breathe. Oxygen is really bad for a lot of processes in cells, but it's also very bad for telomering DNA. And fortunately, we have a great system in our body by which we capture the oxygen and hold it so that it can't do any harm, and then we release it where it's needed. It's called your red blood cells and hemoglobin. So oxygen is a major danger. Other than that, I don't know of an obvious danger.
No lifestyle variables, right?
No.
Many people are, when they have accidents or something like that, they go into oxygen chambers. What effect does an oxygen chamber have on the human being?
I have no idea. I really wouldn't know. I mean, your system is, you know, it's very good in keeping the oxygen level in your tissues low, so it shouldn't really change that much, but I have no idea.
Thanks so much, Titia. You had mentioned that in ascertaining the loop format, that was done with the help of electron microscopy.
Yeah.
My question is, what kind of technologies do you need to do your science today?
Well, right now I really need cryo-EM because my little friend Shelterin that I drew out there, that's just a cartoon, but I don't know what it looks like, and I need to know what it looks like. So I'm collaborating with the wonderful Tom Waltz next door to me, and I hope with sufficient generosity we'll have enough cryo-EM microscopes to also accommodate my experiments on Shelterin.
In our new world of gene editors, we're focusing, are gene editors focusing on the telomeres? And if they are, where are they focusing?
Gene editing, the CRISPR technology, is an incredibly useful tool in the lab. It's not yet that useful in the clinic, but we're all confident that will come one day. But most of the labs here at the Rockefeller use CRISPR technology to change genes in cells that we work on so that we can ascertain the function of that gene. My lab does it all the time. We cut the genes that encode for shortening proteins, and we can see what goes wrong in cells. We cut the genes for other important factors that help us dissect how the system works. But to cut in the telomere itself, not that useful. We have done it, and we've learned some interesting things that are probably not of interest to you, but very much of interest to me. But you know, it's not going to get you very far. You're just cutting in a telomere. You could change the letters, and that has been done a long time ago in various organisms, and things go terribly wrong. If you change the letters, shortening doesn't bind, the loop doesn't form, the telomere doesn't work.
Terrific. Titia, with that, I'd like to draw this to a close. Thank you for an incredible talk. It was just great.